very interested in looking at this compound, which targets TMPRSS6, which essentially is a negative regulator of hepcidin in the body. And we know hepcidin is a major regulator of iron in the body. So we knew that there were opportunities based on preclinical work that we've done to look at patients with iron loading conditions, but also patients with polycythemia vera. So we're very excited about that. So what we did was we performed a phase 1 study. In fact, the phase 1 study is continuing, but we have a substantial amount of information that we were able to share more recently publicly. It's a 34-week study. It's open label phase 1, and it includes patients who meet the World Health Organization criteria for polycythemia vera, all of whom are also phlebotomy dependent. So these are patients that require phlebotomies in order to maintain their hematocrit at levels that are considered safe for these patients, and that is 45% or below. PV patients, because of the viscosity of their blood, are at risk for certain CV events and clotting events, thrombotic events that are reduced. That incidence is reduced if you can keep their hematocrit levels below 45, because their blood is very viscous due to the overproduction of RBCs. So it was designed as a typical phase 1 study, multiple cohorts of ascending doses. We looked at doses of Divesiran, our compound, at 3 mg per kilogram, 6 mg per kilogram, and 9 mg per kilogram. And we were tended to enroll up to 8 patients per cohort so that we would have as many as 24 patients at the completion of the phase 1 study. So the doses that we studied, as I mentioned, were given on a 6-week basis. So the regimen that every patient got was 6 doses in each of the cohorts, 6 doses. So week 0, week 6, week 12, and week 18. And then we followed those patients out all the way to week 34. And the reason why we had a lengthy follow-up is this technology, small interfering RNA, is actually quite durable. So we wanted to get a good sense of the effect of the drug. So following the regimen of 4 doses Q6, we followed those patients all the way out to 34 weeks. So what's very interesting is what we were aiming to do was reduce the requirement of phlebotomy. So all these patients, as I mentioned, were phlebotomy dependent. So in their past 6 months, they had to have a minimum of 3 phlebotomies over the course of that 6 months prior to treatment, or a minimum of 5 phlebotomies in the previous year. So that follows the WHO criteria for these patients. And what we were looking for is a reduction, if not an elimination, of the need for phlebotomy by maintaining with the medicine hematocrits below 45, because when you hit 45 or above, that is typically the treatment trigger, if you will, for a phlebotomy. So that's really what we were hoping to see. What we did see, and by the way, I should step back just for a second, we included patients across a range of baseline hematocrits in the study. So we didn't just target patients who were well controlled and look to see if we could keep them well controlled. We also looked at patients whose hematocrit levels were quite high, and some of them were very well into the 50s, so quite high. So we wanted to explore the effect of the drug across a range of hematocrits. What we saw was for the patients who came in well controlled, so for those patients who started on the drug with hematocrits at 45 or below, none of those patients over the course of the treatment required a phlebotomy. So a very robust effect, 100% essentially suppression of the need for phlebotomy in those patients. And in those patients who came in with elevated hematocrits, well elevated, we also saw a reduction, a substantial reduction. And to give you an idea, if we simply count the number of phlebotomies in the previous 6 months for all the patients in the study, and then looked at the number of phlebotomies once they started the drug, we went from 59 phlebotomies in the previous 6 months to just 2 phlebotomies in the period of treatment. And by the way, the study is ongoing, so I'm only talking to that point in time. We'll complete the study over the course of the next, the end of the year, and we'll present the full data results. But the bottom line is, we didn't calculate an exact rate in change pre and post, but we see a very, very substantial reduction. The other finding we had was that the hematocrits in the patients who were who started well controlled stayed well controlled, and those who had the elevated hematocrits, we saw a reduction in their hematocrit levels and obviously a move towards controlled levels. So very, very encouraging across the range of patients, regardless of their baseline status. What we also saw was that patients' hepcidin levels, because remember, the drug is affecting hepcidin levels. You're blocking TMPRSS6, which is a negative regulator, allows for the increase in hepcidin. And when hepcidin increases, it should restrict the amount of iron to the blood, to the bone marrow, and therefore reduce the number of blood cells that are being produced, because the bone marrow needs iron to produce red blood cells. So what we saw was, in fact, an elevation of hepcidin, regardless of their baseline level of hepcidin, across the board. And that's very, very helpful, because that demonstrates that we were actually having target engagement. You know, we're targeting the hepcidin pathway. You want to see that you're having an effect on hepcidin. And in this case, we saw a nice elevation within physiological range, but a nice elevation. And clearly that appears to be associated with the clinical benefit that is accruing these patients in the ongoing phase 1 study. The other thing I will say is that we had no serious adverse events. We had no patients stop or be taken out of the study because of a treatment emergent, treatment associated adverse event. So very comfortable with that. We did have, as you would see with probably any subcutaneous injection, you do get, mild cases of irritation, but all of those were, injection site reactions were considered mild, and all of them were self-limiting. So very, very nice safety and tolerability profile.